Hi there. Today we are going to talk about the function of enzymes. And enzymes are special proteins that speed up chemical reactions by lowering the activation energy. The enzyme that we're going to talk about today is catalase, and catalase is found in all living things. In our body, catalase breaks down a toxin called hydrogen peroxide that your cells produce. And if hydrogen peroxide builds up, it can kill your cells and ultimately you. So catalase is found in all living things, and today we're going to look at catalase in liver, hamburger, spinach, potato, and chalk. And ultimately we're going to see if in fact these substances have catalase and how much catalase they have. When catalase works on its substrate, called hydrogen peroxide, it breaks it down, as I said, into water and oxygen gas. And oxygen gas is something that we can see when that reaction happens. We can see the bubbling and we can hear the bubbling. And so we're going to ultimately determine how much, if any, catalase are in these substances by looking at those bubbles produced. To start the activity, I'm going to take our substrate here, which is hydrogen peroxide, and I'm going to measure out about 5 mils for each test tube. Okay, and I over poured a little bit, so instead of pouring it back into the stock bottle, I am going to pour the excess into the sink. I'm going to go ahead and add that to my test tube. So now that we have our 5 mils in each of the test tubes, this is our substrate on which catalase is going to act. I'm going to make a hypothesis. Looking at all of these substances, chalk, potato, spinach, hamburger, and liver, make sure that you make a hypothesis as to which one you think is going to have the most catalase in it and therefore have the most vigorous reaction, have the most bubbles.